Every living thing on our planet is made of cells. In fact, our planet-wide genetic heritage stretches back to a single cell, the last common universal ancestor. This cell was probably a lot different than our own. However, we still carry sequences from this cell in our DNA. Researchers suspect that around 30 modern genes trace back to this last common ancestor. Since that time, some 4 billion years ago, evolution has shaped all life on Earth in an overwhelming explosion of diversity. In this chapter, we're going to dive in past the outer layer of membranes and proteins into the very center of this animal cell. From there, we will journey outward, much more slowly this time, and look at the core structures and molecules that give rise to life. The smallest and most basic unit of life is the cell. All the complex organs and tissues of your body are made of different kinds of specialized cells, from the cells of your skin to the cells of your heart muscles to the complex neuronal cells of your brain. They all work together to make you do what you do. Prokaryotic cells are the simplest kind of cells. They have no real internal organs, and most of their complexity is related to their cell membranes and internal protein. These are all part of the bacteria and archaea domains of life. However, the rest of the tree of life, plants, fungi, and animals, and in fact almost all multicellular organisms, are made of eukaryotic cells. Eukaryotic cells have a nucleus. The word eukaryote comes from the Greek meaning good nut or kernel. As should be evident, eukaryotic cells can take an amazing diversity of forms, even within your own body. Compare the cells of a human with the cells of a plant. These look similar, but they also have radically different structures and are tuned for distinct functions. In this chapter, we are going to take a deep dive into a eukaryotic cell and look at all the components that make it capable of living. We will compare the cells of animals to those of plants and see how they are the same and how they are different. Here in the nucleus of an animal cell, we will begin our journey outward, starting at the very heart of cellular function and moving through the different avenues and compartments. We will understand how the molecules that we met in previous chapters combine to create life. The nucleus is contained within the nuclear envelope. The nuclear envelope is structurally supported by the nuclear lamina. These are intermediate filaments that help to maintain its shape and structure. The only way out of this nucleus is through a nuclear pore. I think there's one up ahead, but before we pass through this pore, we must encounter some of the other molecules and structures that reside in the nucleus. Here we see a chromosome. All chromosomes are bundles of DNA and reside in the nucleus. They are only really completely bundled up during mitosis. When they unbundle, we can see the chromatin that makes them up. Chromatin is DNA that is tightly packed by proteins called histones. As the chromatin unpacks and becomes ready to produce proteins and replicate, we can see that the DNA is unwrapped from the histone proteins, so it is exposed. In the area of the chromatin, we can also see a larger structure called the nucleolus. This is a special area where ribosomal RNA is transcribed from the DNA and used to build ribosomes. Ribosomes are what help convert messenger RNA into proteins. Oh look, there's a ribosome ready to leave the nucleus. Let's follow it. Look up ahead, it's the nuclear pore. This pore is the gateway from the nucleus to the next layer of the cell, the endomembrane system. Awesome, we made it through. We are now just outside the nuclear envelope, but inside what is called the endoplasmic reticulum. The endoplasmic reticulum is a maze-like membrane that is continuous with the nuclear envelope and forms an arch around the outside of the nucleus. The inside of the endoplasmic reticulum is called the cisternae. This here is the smooth endoplasmic reticulum. Sure is a lot of calcium in here. This is where many cell types store calcium that can be used to activate functions like muscle contraction. Besides storing and releasing calcium, functions that the smooth endoplasmic reticulum is responsible for includes drug and poison metabolism, lipid synthesis, and carbohydrate metabolism. Oh, look over there. It's an enzyme breaking down a toxic substance. And over here, there are enzymes which are synthesizing lipids. Let's get out of here before we get broken down. We've not moved far, but now we are in what is called the rough endoplasmic reticulum. This name comes from the fact that the walls of this place are full of ribosomes. Like our friend from earlier, some ribosomes escape the nucleus directly into the cytosolic part of the cell, which we will come to later. However, many ribosomes are trapped here in the walls of the rough endoplasmic reticulum. These ribosomes are responsible for making proteins that will ultimately be secreted in vesicles. Also, glycoproteins are made here. These are proteins with polysaccharides attached to them. Other proteins made by these wall-bound ribosomes will be inserted into the membrane and potentially end up in the outer plasma membrane. They are transported and processed in transport vesicles like we mentioned earlier. Let's pop out of the ER and have a look at this from the outside. Look at that. The vesicle buds off from the rough endoplasmic reticulum and is transported to this amazing structure that we see here. This is called the Golgi apparatus. The Golgi apparatus is like the shipping and receiving part of the cell. Transport vesicles bud off from the rough endoplasmic reticulum and fuse with the cis Golgi. Remember that cis means this side up. Proteins move through the Golgi apparatus, are modified and prepped, and then shipped off in transport vesicles. The way that proteins move through the Golgi is not entirely understood, but it is clear that they move through and re-emerge again in vesicles from the trans side of the Golgi. Each of these cisternae are responsible for different stages of modification. Within the Golgi cisternae, macromolecules like polysaccharides are made. In addition, the glycoproteins that were made in the rough endoplasmic reticulum can have their polysaccharide additions modified here, in the Golgi. Both protein and non-protein products of the Golgi all leave in transport vesicles from the trans side of the Golgi apparatus. These products are all sorted and marked with tags to be sent to different parts of the cell. These are molecular identification tags. Additionally, vesicles themselves have specific docking proteins that will bind to their partner proteins only at specific cellular locations, like the plasma membrane or other organelles. See those vesicles over there? Those are lysosomes. Lysosomes are filled with hydrolytic enzymes that break down food and cellular debris. They are like a floating recycling factory. When they merge with other vesicles, they attack and digest the contents of that other vesicle. The vesicle that it merged with was a food vacuole. We will see how food vacuoles form when we get closer to the plasma membrane. From here, we look around the cytoplasm. The cytoplasm is the main compartment of the animal cell and is filled with cytosol. The nucleus, endoplasmic reticulum, and Golgi apparatus are below us. We can see free ribosomes making proteins that stay in the cytosol to do work, like glucose metabolism. What we see in the background is the cytoskeleton. The cytoskeleton is made of 3 basic types of proteins. These proteins work together to maintain cellular shape and provide a means of moving packages from one place to another using motor proteins. The first of these are microtubules. Microtubules form when small proteins called tubulin bind into dimers. That is a protein made of 2 parts, which then gather into a long, hollow, straw-like structure. These structures serve a variety of functions in the cell. They are here to help maintain structure, and as a railway for motor proteins to move things around. Microtubules generally originate from the centrosome. The centrosome is a pair of centrioles which serve to organize microtubule formation in animal cells. Some cells lack a centrosome completely, and the microtubules are organized in different ways. During cell division, microtubules are critical for getting things sorted into their proper place. Over here, we see our next cytoskeletal element, microfilaments. These are long, solid rods made from lots of smaller proteins called actin. The microfilaments are there to bear the weight of the cell and keep it in shape. They can also be rearranged if the cell needs to move. We will look more at that later when we talk about flagellum. Microtubules and microfilaments can be quickly assembled and disassembled as part of cellular rearrangement. However, our last cytoskeletal element is much more permanent. The last group of filaments are called intermediate filaments, and, like the other 2, are made of smaller proteins. Intermediate filaments are made of a class of protein which includes, but is not limited to, keratin. Intermediate filaments help hold the cell shape and keep organelles in place, but they remain relatively stable and do not participate in cell rearrangement or movement. See that dark shape in the distance? That is our next stop, and it is an incredible organelle called the mitochondria. Mitochondria are a double membrane organelle that help produce cellular energy. The more active a cell, the more mitochondria it'll have. Here, we are at the heart of the mitochondria, inside what is called the mitochondrial matrix. We see ribosomes floating around whose job it is to make mitochondrial proteins from, you guessed it, mitochondrial DNA. Mitochondria have their own DNA that is separate from the DNA normally found in the nucleus of the cell. From up above, we can see that the inner membrane of the mitochondria is folded into what are called cristae. On the other side of the cristae is the inner membrane space. This is the gap between the outer smooth membrane and the inner folded one. If we look closely, we can see proteins bound into the inner membrane. These are proteins involved in making ATP. The metabolism of glucose and other molecules helps drive this reaction, and much of that happens here in the mitochondria. Mitochondria can divide, which is called budding, and can merge together to form large mitochondria. Now that we're back outside the mitochondria, we can make our way to the plasma membrane. We can get there by following this microtubule, like motor proteins do. Motor proteins use ATP to move along the microtubule. The motion is very much like a walking man. These motor proteins are called dyneins. They are basically a porter who's carrying attached vesicles to different locations around the cell. We're almost there, but let's stop for a second and look at this vesicle passing by. This vesicle is a peroxisome. Peroxisomes contain enzymes that convert harmful oxygen radicals into hydrogen peroxide. Hydrogen peroxide would be toxic to the cell if it wasn't sequestered here in the peroxisome. This is a great example of a connection between structure and function. Here we are at the outermost barrier of the cell, the plasma membrane. We can see that all kinds of cytoskeletal elements and proteins are connected to it. We see some of the vesicles from the trans-Golgi have made it this far, and are fusing with the membrane. Their contents are being expelled in a process called secretion. Additionally, proteins that were in the membrane of the vesicle are now part of the plasma membrane. This is how proteins get from the rough endoplasmic reticulum into the plasma membrane. The plasma membrane is less a barrier to the outside world and more a regulatory interface. Substances are brought in by the invagination of vesicles. These can form things like food vacuoles. Additionally, substances can be expelled by the fusion of vesicles like we saw in the process called secretion. Also, the proteins in the plasma membrane regulate which molecules can come in and out of the cell. Even still, nonpolar molecules are able to pass right through the membrane directly. This is called diffusion. We will discuss the details of the plasma membrane more in the next chapter. For now, we can just be content to understand that this membrane is an interface to what is outside the cell. In this way, all life is directly connected to and a product of its environment. This massive structure here is called a desmosome. Desmosomes are one of the three important sets of proteins that help connect the cells of multicellular organisms to each other. They are desmosomes, tight junctions, and gap junctions. These desmosomes are anchored to the cytoskeleton with intermediate filaments made of keratin and help keep cells tightly bound. Similarly, tight junctions are a set of proteins that bind cells together so tightly that fluids and other molecules can't pass between them. Gap junctions are circular collections of connexin proteins. They bind to similar structures on adjacent cells and can regulate the flow of small molecules between the bound cells. Gap junctions are known for allowing large groups of cells to become electrically coupled and work together to produce coordinated muscle contractions, like in the heart. Let's pass through this transporter protein and emerge outside the plasma membrane. From here, we can see that there are as many different molecules holding things together out here as there were inside. This is because cells are tied to their environment. They are an extension of everything around them. What we see out here is called the extracellular matrix. We see collagen, which makes up about 40% of the total protein in the human body. These proteins are embedded in a network of proteoglycans. In addition, proteins that link to the inside of the cell are called integrins, and these bind to fibronectin on the outside of the cell. This allows a direct link between signals from the extracellular matrix and the inside of the cell. Again, we see the interdependence of the cells on their environment. This link between us and everything around us is one of the most amazing facts of nature. The biosphere is a tapestry of interconnections between living cells and their environment. Neither would be what it is without the other. This understanding is critical to the survival of our species and of the biosphere. Now that we have taken a tour of the typical animal cell, let's review some of the things we saw. First, we saw the nucleus, which is a membrane-bound space held in shape by intermediate filaments. Next, we saw the smooth and rough endoplasmic reticulum, where proteins and lipids are made and toxic substances are broken down. This leads us to the Golgi apparatus, where proteins and molecules made in the endoplasmic reticulum are sorted and shipped to targeted parts of the cell. We then looked at the cytosol, the main body of the cell which contains other organelles, such as the mitochondria. We finally looked at the plasma membrane and all the filaments, tubules, and proteins that serve as the interface between the cell and the outside world. We saw how vesicles fuse with the membrane, and discussed how things like food vacuoles form when substances are taken up by the cell into vacuoles. In general, vacuoles are larger than vesicles, and they don't fuse readily with other larger membranes like vesicles do. Next, we looked at the cytoskeleton. As we saw, these filaments and tubules are connected to both the internal and external parts of the cell. They're also capable of being rearranged, which results in cell movement, and they allow for structures like the flagella and the cilia to propel single-celled organisms through the environment. We are animals, which can make us think that animal cells are the default. However, plants and photosynthetic organisms had to evolve long before animals because they are the animal's food source. Here's a diagram of the difference between a plant cell and an animal cell. We see that both have a lot of the same structures that we've already looked at. They both have a nucleus, an endoplasmic reticulum, Golgi apparatus, mitochondria, cytoskeleton, and a plasma membrane. They also contain lysosomes and peroxisomes. However, there are some differences here that are important to look at. First, we notice that the plant cell has a central vacuole. This is a large membrane-bound structure right in the middle of the cell. This massive vacuole functions as a storage container that can also break down waste products and perform hydrolysis of macromolecules. In other words, breaking down polymers into smaller polymers and to monomers. Additionally, plants have a cell wall. When a plant is young, it secretes a matrix of proteins and cellulose to form the primary cell wall. This wall protects the cell from taking in too much water and helps to support the structure of the plant. Between this wall and the walls of the neighboring cells are what is called the middle lamina, made from sticky polysaccharides called pectin. This middle lamina helps keep the cells stuck together. As plants age, they sometimes add what is called a secondary cell wall between the plasma membrane and the primary cell wall. This helps to strengthen the plant structure. These cell walls also contain membrane-lined channels that can pass water and small molecules between the cells. These channels are called plasmodesmata. On our way through the animal cell, we looked at the mitochondria. We saw that the mitochondria was a double membrane organelle, which means it has an inner and an outer membrane. Plants have mitochondria as well, but they also have another double membrane organelle called the chloroplast. It is thought that both of these organelles were once free-living organisms that got taken up by the ancestor of eukaryotic cells. They both contain their own DNA and ribosomes, and they both offer functions that the main cell is not capable of without them. Mitochondria produce large amounts of ATP energy from glucose and fats, and chloroplasts convert light energy into chemical energy. The theory that these organelles were once free-living and made a home inside of an ancestor cell with a nucleus is called the endosymbiont theory. Chloroplasts are a double membrane organelle with an outer membrane, an inner membrane space, and an internal compartment that contains an interesting system of interconnected, flattened sacs called thylakoids. One stack of these sacs is called a granum. The thylakoids are surrounded by a fluid called the stroma, which also contains all the chloroplast DNA and ribosomes, as well as a variety of enzymes. And similar to the mitochondrial matrix, the thylakoid membrane contains proteins that use the energy from light to produce ATP. From the nucleus of the cell to the outer membrane and even the cell wall, all cells are connected to the environment. Every cell on Earth is an extension of all the chemical processes on the planet, and thus are connected to each other in this grand scheme of interactions we call the biosphere.